Hi, welcome to this next video tutorial in your general biology course. This session is entitled Gene Regulation. So we've talked quite a bit about how genes get transcribed into messenger RNA and translated into proteins. What we want to do now is focus on how those genes are regulated. In other words, how does a cell know when to turn on and turn off genes, and how does that actually happen? So let's go ahead and take a look at our learning objectives. The first thing we're going to talk about is the Human Genome Project. This was a massive data gathering activity where we discovered which genes are actually present in the human genome. And then we'll get into our discussion of gene regulation, and we're going to focus specifically on how genes are regulated in eukaryotic cells. Then we'll talk about DNA microarrays. This is a research tool that scientists use to find out which genes are in fact turned on or off in a cell at a given time. And then finally we'll talk about some applications of this gene regulation concept as it pertains to stem cells and cloning. Alright, so again, all semester we've been asking this question, what is life? We've been working our way through these 6 different features of life. And in this video, we're going to continue our discussion of growth and development and regulation for that matter. After all, we're looking at how genes get turned on and off during the process of growth and development. So to get started, we're going to define a couple of terms that actually refer to pretty new fields of biology, really new within the last decade or so. The first field of biology is called genomics. So as you can see with the word gene at the beginning of that, this involves studying genes and DNA. In fact, it is the systematic study of whole sets of genes and other DNA sequences and their interactions within an organism, as well as genome comparisons between species. In other words, in the field of genomics, what we're trying to do is identify all of the genes that are present in an organism's genome. We also know that there's more than just the genes in the genome. There's the regions of DNA in between the genes that play an important role, some of which we'll see in this video. And then we want to compare the genes and regulatory sequences of one species to another. Because although we haven't gotten into it yet, that's going to help us understand the evolutionary process in a lot more detail. Now, a corollary to genomics is a field called proteomics. So again, you see the root word proteo implying proteins. Genes, of course, encode proteins, or the recipes for proteins. So proteomics is really an extension of genomics. This is the systematic study of sets of proteins and their properties, including relative abundance and interactions with other proteins. So we know that genes encode proteins, but proteomics looks at what are the proteins, what is the structure and role served by that protein in a cell, and for that matter, how much of that protein is made and when is it made, and do those proteins interact with other proteins. So these two fields collectively are really giving us a huge picture of what's going on inside a cell. Now, in order to do genomics, you would have to know which genes are in fact present, and although DNA sequencing methods have been around for a little bit, it's only been in the last decade or two that we've had the ability to sequence giant genomes, such as the human genome. And so that leads us into a discussion of the Human Genome Project. So humans, of course, are some of the most complex organisms on Earth, and we have a very large genome as a result of that. In fact, scientists speculated that humans would have a few 100,000 genes. And so it was believed it would take an enormous amount of time to sequence the entire human genome. Well, it seemed valuable enough to pursue it, though, so a lot of research universities along with the U.S. government decided that they would attempt to do it. And in fact, different research institutions were assigned different regions of the genome. You know, Stanford, you guys take this little region of this chromosome, and Harvard, you guys take this region, and this national lab, Oak Ridge, you guys take this region, and we'll just pool all our data together at the end. And the person who was tasked with overseeing all of that was Francis Collins. But meanwhile, a private researcher in the private sector, Craig Venter, had developed some new sequencing technologies that he thought were faster, and he decided he was going to form a company to sequence the Human Genome Project on his own. And he stated that he would get it done faster and cheaper than the government-sponsored project. Well, that didn't make any sense to the president at the time, which was President Clinton, to have two different groups undergoing the same project. The limitations on the government version of the project is that it would take a long time, but it would be extremely accurate. Craig Venter's company that was going to do it could do it much faster, but with less accuracy. And ultimately, we needed the best of both worlds. So President Clinton made Francis Collins and Craig Venter sort of work together. Neither one exactly wanted to, but he forced them to sort of work together. And as a result, it turned into a very efficient collaboration of private and public sector research. And they did, in fact, get the genome sequenced faster, cheaper, and with all of the accuracy that was necessary. So let's look and see what we learned from the Human Genome Project. The first draft of it was published right around the turn of the century. So the most startling thing that we learned from the Human Genome Project is that humans have about 22,000 genes. This was way lower than the estimate that scientists had going into the project. Again, we thought we would have a couple 100,000 genes as a reflection of just how complex we are as organisms. So scientists were very surprised to find that the number was only 22,000 genes. And once we had the technology and the methods for sequencing improved, scientists sequenced the genomes of lots of other organisms and began comparing those genomes to the human genome. So here's a picture of some of the things that have been sequenced. Humans, as we said, have 22,000 genes, but let's now look at the chimpanzee and the mouse and that Cinerabidus elegans microscopic worm and plants and bacteria, and here's some of the things we have found. First of all, chimpanzees and mice essentially have the exact same 22,000 genes as humans. That was also a very startling discovery. Again, we think of humans as being so complex, we thought our genome would reflect that complexity and be very different than the genomes of, say, mice or even chimpanzees for that matter. And even that little common roundworm, Cinerabidus elegans, has 21,000 genes, many of which it shares in common with humans. So the results of the Human Genome Project required us to drastically rethink our genetic paradigms in terms of how genes work and function. So the question became, how can two organisms have such different phenotypes and yet such similar genotypes? How can the same set of genes, essentially, the 22,000 genes, result in brains that compose symphonies and brains that hunt for cheese? It really was a puzzling question. And what came of that is really a shift in our understanding of the genome itself. When we looked at it a little bit more closely, it seemed to make more sense that humans and mice might have a lot of genes in common. After all, they make a lot of the same proteins. Humans have hair, mice have hair. Humans have skin, mice have skin. Humans need hemoglobin for blood. Mice need hemoglobin for blood. We both need collagen. We both need digestive enzymes for the digestive tract. So in many ways, the proteins are pretty similar. And so what we've discovered is the real difference between the phenotypes of humans and mice is not a matter of which genes you have. It's a question of which genes are expressed and in what combination at what time. The reason you see a keyboard in the back of that picture is because that's the metaphor I want you to think about. Think about the set of 22,000 genes as being a bit like a keyboard. A piano keyboard has 88 keys. You can play 88 different notes. Kind of like a genome with 22,000 genes can play 22,000 different proteins. So the difference between Bach and Beatles and Beastie Boys and Beyonce and Black Eyed Peas is not a matter of the keyboard. It's a question of which keys, which notes are being played at what time and what combination, and then chronologically the pattern that plays out over time. In other words, the difference between mice and humans is more about the sheet music, the instructions, the guidance, the orchestration of the genes that's determining which genes are being expressed at what time and in what combinations. So what we really need to focus then on in this video is gene regulation. How does the cell know which genes to turn on at what time and in combination with which other genes? Now, interestingly, we've already discussed this a little bit. We got into this in a previous video when we talked about prokaryotic gene regulation. In other words, how genes are regulated in bacteria cells, for example. And if you recall, Jacques Monod had figured out something called the operon model of gene regulation. So this is how bacteria do it. And let's just remind ourselves real quick of what an operon was and how that works. So if you recall, an operon is a functional genetic unit consisting of genes and regulatory sequences that control the expression of one or more genes. And if you recall from our video, we talked a little bit about the lac operon. As we begin to think about eukaryotic gene regulation, however, it's more complicated. There's a lot more steps involved, which also gives us a lot more places to perform the regulation, so they can be more highly regulated because it's a more complicated process to begin with. So what we'll see is that there are several features or steps to eukaryotic gene regulation. First of all, cell signaling will be involved. Some sort of a signal will arrive at the cell to tell it to turn on or possibly turn off a particular gene. And then, once the signal has arrived, the chromatin, the genetic material, may need to be modified a bit to make genes accessible for transcription. Recall that chromatin's pretty compact. We'll look at it more in a second here. And we've got to unravel some sections just to make the genes available for transcription. And then we'll see it's not as simple as just polymerase binding to the promoter. We will need some transcription factors to help initiate transcription of the gene. And then once the RNA has been transcribed, we'll introduce a concept called alternative mRNA splicing. The RNA itself may need to be processed a bit before it can be sent to the ribosome for translation. And then, en route to the ribosome, we will encounter something called microRNAs, little tiny RNAs that can bind to messenger RNAs and either destroy them or block their ability to get to the ribosome. And then finally, if an mRNA is translated into a protein, that protein may still need to be processed in order to do its job. It may need to be phosphorylated to be activated or something. And proteins degrade over time. So we'll need a way to regulate that process and get that gene turned back on again if necessary. So those are the steps we need to work through. So let's start with the first one, which is cell signaling. And this is a topic we've already discussed in this class. So here's just a reminder of how cell signaling works. If you recall, there's 3 steps: reception, transduction, and response. So let's say some sort of a signaling molecule shows up, maybe a growth hormone or something shows up, binds to the receptor, the signal is relayed or transduced to the cell, and then the cell responds. And specifically now, we're going to be talking about a nuclear response because we're going to be turning on or off a gene. So to get the gene turned on or off, many times that begins with some sort of a signal molecule. Some genes get turned on or off without requiring a cell signaling step. It's all done internally in the cell. But often there's a cell signaling step involved to get the cell to respond in a certain way. So let's assume that the signal has arrived and the cell is being told to turn a gene on, sort of to play a note on the keyboard. Well, here's the steps that are going to take place inside the cell for that to happen. They're shown here in multiple steps. And that figure can look a bit confusing. There's a lot going on in there, but we can summarize it with this figure here. So once the signal has arrived, the first thing we're going to have to do is modify the chromatin to make the gene accessible. Once the gene is accessible, it will be transcribed into the RNA, but then that RNA may need to be processed. We'll talk through that. It eventually does leave the nucleus, however, and is en route to the ribosome for translation. But we'll see that that messenger RNA may be destroyed or degraded before it even gets to the ribosome. So that will be a possibility. And then finally, once the protein has been made, it may again have to be processed in some way before it can do its job, and it might be degraded over time. So those are the steps we need to work through. And the advantage, as far as a eukaryotic cell is concerned, is that each one of these steps is kind of like a knob that we can turn and adjust and control. So look at all these various steps of regulation that the eukaryotic cell has at its disposal. So it's not just as simple as turn on a gene, make a protein, and we're done. There's all these little controls in there that can help regulate, well, when is the gene turned on? How much mRNA is made? How is that spliced? When does it get to the ribosome? How much protein is produced? How long does the protein last? So we've got lots of places where we can regulate this. Well, let's begin with chromatin modification, since that was the first step here. Let's remind ourselves for a second what chromatin is. The word literally means colored material, and we've talked about this quite a bit already this semester. So it is the genetic material. It's a mixture of DNA and proteins. The DNA in this picture, of course, is that blue double helix structure. The proteins are those little purple balls, and they all get compacted together into threads that get compacted further into the chromosomes. So there's a lot of compaction going on to make sure all this fits in the cell. So these little proteins are called histone proteins, and the DNA wraps around little clusters. You can see it takes about 8 of them to form a little cluster, and the DNA wraps around that to form little units called nucleosomes, or nuclear bodies. And then the nucleosomes, each one of those little 8 histone with DNA wrapped around it clusters, the nucleosomes get all compacted together to form a strand, which then gets coiled even further and compacted into the chromosome. So there's a lot of stuff going on there. And of course, along the way, we have the genes, regions of the DNA that encode particular proteins. But as you can imagine from this picture, those genes get buried within the nucleosomes and within all that compacted DNA in the chromosome. So in order to make a gene accessible for transcription, if the gene needs to be turned on, the histone proteins may need to be acetylated, adding acetyl groups. We don't need to get into the depth of chemistry here, but it's a 2-carbon functional group of a thing. So acetylation of the histone proteins can loosen the coils and make genes accessible for transcription. In other words, allow the gene to be turned on. So let's look a little more closely here. So you can see the histone proteins have those little sticks poking out, and on the sticks have been placed acetyl groups. And what that does is causes the nucleosomes to separate from each other, creating the spacing in between. And notice that's where the genes are located. So by acetylating the histone proteins, the chromatin becomes uncoiled a bit, making the gene accessible for transcription. So we really can't transcribe the gene unless the gene itself is exposed and available. Now, interestingly, there's the opposite thing that could be happening. DNA can become methylated, that is, add methyl functional groups to it, and the nucleosomes remain compacted together. So in this scenario, the genes remain kind of hidden and buried in there and are not available for transcription. So if a gene is going to be turned on, if we're going to play the note, one of the first things that has to happen, perhaps, is the chromatin modified to make the gene accessible. Now, these two ideas, acetylation of the histones and methylation of the DNA, are things that we call epigenetics. So these are things that are added on top of the genes, literally epigenetics, acetyl groups and methyl groups, that help regulate when and where a gene is expressed in a cell. And this was a pretty interesting discovery in the last decade or so. So epigenetics is a new field of study. It is the study of heritable phenotype changes that do not involve sequence alterations in the DNA. We have previously always assumed that the only way to have a particular phenotypic trait is to have a gene for that trait. Well, now we know there's more to it than that. Just because you have the gene for the trait doesn't mean the gene will be expressed, because these epigenetic patterns of acetylation and methylation can control whether the gene is expressed at all. So we could get into ideas like having predispositions for traits, but not necessarily showing the trait. And what's interesting is these epigenetic patterns, in other words, which genes are methylated and which ones are acetylated, are influenced by our behaviors and our lifestyle choices. In other words, things we do can impact our genes and dictate, or at least influence, whether genes are expressed or not. And we have traditionally assumed that we're sort of at the mercy of our genes, whatever genes we have, but now we're discovering that our behaviors and lifestyle choices can actually influence how those genes are utilized, for better or for worse. In fact, I'll show you an example that is actually for worse. So it turns out that a woman who smokes while pregnant, and that would be a behavior or a lifestyle choice, a woman who smokes while pregnant induces epigenetic changes in 3 generations at once, in herself, in her unborn daughter, and in her daughter's reproductive cells, which would influence the 3rd generation there. And so smoking is a choice that you really should avoid while pregnant, because that choice is going to affect the methylation and acetylation patterns of not just your DNA, but of your baby's DNA and of your baby's reproductive cells, so that when your baby grows up and reproduces, the next generation is impacted as well. So this one person's choice is going to impact 3 generations of people. And one of the things that cigarette smoking while pregnant has been linked to is an increase in the likelihood of obesity. So let's see what happens. Here is our pregnant mother who is smoking, and that is not only affecting the epigenetic markings of her DNA, it's influencing the epigenetic marking patterns of her baby, but also of her baby's reproductive cells. And we're going to assume the baby's a daughter in this picture. So her baby's shown in red, and then her baby's reproductive cells are shown in blue. So they're forming right away here at the time the fetus is forming as well. So now the baby is born. That's the next generation. And unfortunately, this baby is born with some of those epigenetic markers of the mother, and that increases the likelihood of obesity in this person. But recall that her reproductive cells were also impacted, and so when she has a baby, these epigenetic patterns are being passed on, and so that ends up in the 3rd generation. And notice that the 3rd generation is at an even greater risk of obesity. So we're seeing the likelihood of obesity increase across 3 generations because of the smoking that was taking place with the mother at the very beginning of the story. So epigenetics is really a fascinating area of study that we're just now starting to study in more detail. Now, I used an example there of a woman's behavioral choices causing epigenetic changes, but the same holds true for males. When males produce sperm cells, those sperm cells can carry some of the epigenetic markers of their behaviors and lifestyle choices as well. All right, well, let's move on from chromatin modification to the next step, which is transcription. Let's assume that the chromatin is modified in some way. The gene is accessible. It's available for transcription. So now we want to look at what unfolds next. So transcription, we've talked about this quite a bit. That is where RNA polymerase binds to a promoter and transcribes the gene into mRNA. But it turns out there are transcription factors that need to be in place to allow that to happen. So transcription factors are substances that bind to enhancer sequences and facilitate the binding of RNA polymerase to the promoter sequence. So let's take a look here at a region of DNA. You can see to the far right is the gene in solid blue, kind of going off the screen. Right next door to it is the promoter. But we want to focus here for a second on these enhancer sequences. So these are where transcription factors are going to bind, and that's going to help RNA polymerase bind to the promoter and get the gene transcribed. So normally RNA polymerase should bind to the promoter, but it turns out that RNA polymerase really has a very low affinity for the promoter sequence. In other words, it doesn't bind very easily to the promoter. So by default, really transcription's not going to occur unless we have these transcription factors. And what they do is they bind to the enhancers, And that causes the DNA to bend around and recruit the RNA polymerase to the promoter. And we can see that in a picture. So notice how the transcription factors have bound to the enhancers and form this little complex that pulls in the RNA polymerase to the promoter. So again, that's literally the definition we see on the screen. Transcription factors are substances that bind to enhancer sequences and facilitate the binding of RNA polymerase to the promoter sequence. RNA polymerase will not bind to the promoter just on its own. It needs the help of these transcription factors. So what's the point of having transcription factors? Well, it allows for differential gene expression in different cell and tissue types. And let's talk about what that means for a second. Here is a set of 5 different genes. Now recall that every cell in your body has the exact same set of genes. It's the genes you got from your parents. So whether it's a skin cell or a liver cell or a kidney cell or an eyeball cell, every cell in your body has the exact same set of 22,000 genes. However, each cell has different types of genes being expressed in different combinations and times. In other words, this is the sheet music. Each cell has its own little unique song or music that's being played in there about which keys are pressed at what time and in what combinations. So we need a way to express different genes in different cells, and we call that differential gene expression. And what's going to allow for this are these enhancer sequences that each gene has. Now notice that each gene has a slightly different enhancer region. So the colors are referring to a little sequence there where a transcription factor can bind, and the color tells us what kind of transcription factor is necessary. So if we look at gene 1, we need an orange transcription factor to bind, a blue one to bind, a green one to bind, and a black one to bind. And if all those transcription factors show up and bind to those enhancer regions, gene 1 will be turned on. Meanwhile, this gene, number 2, has a slightly different transcription factor requirement. We need some sort of black transcription factor, blue factor, and green factor to come in and bind to bend this around and get the gene expressed. And yet this one's different. We need the orange, the blue, and the green factors to show up. So it's almost like each gene has its own little unique barcode. And if the right transcription factors show up, then that gene will be transcribed. Now let's look at a slightly more specific example. So here are 2 different genes that you have in your body, the albumin gene and the crystalline gene. Albumin is a protein, and this is the gene that encodes it. Albumin is a protein that needs to be present in liver cells. Crystalline is a protein that is needed to be made in eye cells, particularly to produce the lens of the eye, the crystalline proteins that form the lens of the eye. So the albumin gene and the crystalline gene are both present in a liver cell, but we only want the albumin gene to be on. Likewise, in an eye cell, both genes are present, but we only want the crystalline gene to be expressed there. So how can we get these different genes expressed in different tissues? Well, we're going to rely on transcription factors and the fact that the 2 genes have slightly different enhancer regions. Here's the enhancer region for the albumin gene. We need some sort of a yellow factor to come in here, a gray factor, and a red factor, whereas the crystalline gene requires a gold factor. I'm using colors just to tell them apart. They're really substances, but we need the gold factor, the gray factor, and the pink factor. So it's interesting, they do share the gray enhancer region in common, but otherwise their enhancer regions are slightly different. Well, let's go into the 2 different cell types, the liver tissue and the eye tissue, and see how this works. So let's start with a liver cell. So notice in the liver cell, the red factor, the gray factor, and the yellow factor are present, and so they bind and they help recruit the RNA polymerase to the promoter, and the albumin gene is expressed. But the crystalline gene is not expressed. The gray factor is present, We needed that for the other gene, as you can see, but the gold factor and the pink transcription factor are not present, so that gene is not expressed in liver, exactly the way we want it to be. Meanwhile, if we go to that eye, we see something a little different. So in the eye, we have the pink factor, the gray factor, and the gold factor. Those factors are present in lens cells, and so they get the RNA polymerase recruited to the crystalline gene and it's turned on. But we do not have the correct transcription factors present in that cell type to turn on the albumin gene. So again, we're getting differential gene expression in different cell and tissue types. All right, so we've talked about getting the gene transcribed. We need the right transcription factors to ensure the gene is transcribed. And that, of course, results in RNA. But now we need to talk about RNA processing. So we don't just immediately get messenger RNA, it turns out, from transcription. So let's look at something called alternative mRNA splicing. This does not occur in every case. In fact, many genes do not involve alternative mRNA splicing. But some do, and so that's why we want to talk about what this is, because it's a pretty fascinating concept with some interesting implications. So it turns out that some genes consist of exon regions or express sequences separated by intron or intervening sequences. So if you look at the gene down there, you see the yellow exon, the green exon, the orange, the purple, the red. These are not transcription factors or enhancers or anything. This is the gene itself now. This is the stuff that encodes the protein. The exons are sequences that will ultimately be expressed, meaning translated into protein. But we need to think about these intervening intron sequences, which are shown in blue. So the blue regions in between are called introns, and these regions are not expressed. In other words, they're located in the gene, but they do not contain information that is supposed to be translated later into a protein. Nonetheless, both the exons and the introns get transcribed into what we'll now call a primary RNA transcript. So I know in previous videos we would have said transcription, you produce messenger RNA. But we've got this middle player here, the primary RNA transcript. It's going to become messenger RNA, but not just yet. So the gene gets transcribed, exons and introns, everything gets transcribed, and we get this primary RNA transcript. But now what we need to do is remove those intervening intron sequences. So the intron sequences are removed, and the exon sequences spliced together to produce the final messenger RNA transcript. So we made this temporary version that's got the intervening sequences. We need to cut those out and glue together the exons. But as we're going to see, we have some options as to how the exons are glued together. So let's take what we've seen so far, move that to the top of the screen there. So we've already discussed what's going on there. And now we're going to do what's called alternative splicing. We're going to remove the blue intron regions and start to put together the exon regions. But we can do that in a couple of different ways. One way would be to put exon 1 next to 2, next to 4, 5, 6. Notice we left 3 out, so we might even remove an exon as well. So that's one way to splice those exons together. But another way to splice them together might be 1, 3, 5, 6, or it might be 1, 3, 4, 5, 6. In other words, it's almost like shuffling the cards in a deck. You can rearrange the exons when you glue them together to be the messenger RNA. And for that matter, you can eliminate some, like we did. So why would this make a difference? Well, if we now translate these, if these messenger RNAs go to the ribosome to be translated, we're actually going to get different kinds of proteins, because those exons refer to different sequences, different types of codon arrangements. If we translate the first one, we get a protein that kind of looks like this. But notice the second option there, the 1, 3, 5, 6, that would be translated into a protein that's built a little bit differently. And the third one would be yet a third version of a protein. So we've got essentially 3 different versions of a protein, all potentially coming from the same single gene. In other words, one gene is encoding multiple versions of a protein, or has the potential to encode multiple versions of a protein. And so this shuffling and recombining of exons allows for a single gene to encode several variations of a protein. Now, unfortunately, in this class, we're not going to be able to get into a lot of the implications of this, but if you were to take a course in immunology, for example, you would see that this really explains how we produce the billions of different shaped antibody proteins that we use to defend ourselves against pathogens. Also, this idea that one gene can encode different versions of a protein or almost experiment with different kinds of proteins plays a very important role in the whole evolutionary process. So the take-home message here is that even though a gene gets transcribed, there are alternative ways to splice together the messenger RNA, ultimately determining what kind of protein is produced from it. And again, not every gene involves alternative mRNA splicing. In fact, most probably don't, but some eukaryotic genes do involve this splicing process. All right, so let's go back to our list now. We've modified the chromatin, we've gotten the gene transcribed, the RNA has been cut and pasted together correctly. It is now headed to the ribosome to be translated. But it may encounter something en route to the ribosome that we need to talk about that could result in mRNA degradation. So those things are called microRNAs. So microRNAs are small RNA molecules that can block or inhibit translation by binding to messenger RNA molecules. So just because a messenger RNA gets produced doesn't necessarily mean that it will make it to the ribosome to be translated. These microRNAs may get in the way of that. So let's see how that works. So here is a microRNA shown in yellow. They're little single-stranded stretches of RNA, not that many nucleotides long. They stick themselves inside a protein, so they do their job inside a protein. And they wait to see if a messenger RNA shows up that is complementary to them. And if a messenger RNA shows up, some things might happen. So the first scenario is, let's say a messenger RNA shows up, that's what's in pink. That messenger RNA was headed to the ribosome, but it kind of got stopped by this microRNA that turns out is complementary completely to at least that region of the messenger RNA. So it only binds to a short region, but we notice that every nucleotide of the microRNA is complementary. It's kind of a complementary fit in every location. And if that's the case, the messenger RNA will be destroyed, as it turns out. So complete complementary binding of microRNA to messenger RNA causes the mRNA to be degraded, and so it won't be translated at all. We went to all that trouble to get it transcribed, but now it's destroyed before it ever gets to become the protein. So the other scenario is that we get partial binding. So now in this scenario, the microRNA matches the messenger RNA in most of the locations, but not all. You see a little region in the middle where apparently the G's and C's and A's and U's don't agree completely with each other. So we're not hydrogen bonding there in the center. We get that little bulge. Well, if that's the case, the mRNA is not destroyed, but is at least inhibited and delayed in getting to the ribosome. So incomplete binding prevents the mRNA from entering the ribosome. So that's blocking translation, at least temporarily. So those are microRNAs. Let's go back now and let's assume that the messenger RNA does get translated eventually and we produce a protein. So we've made it all the way from turning the gene on, transcribing it into RNA, getting it to the ribosome, getting it translated into protein. Now we still have a few knobs left. The cell has a few ways to regulate things. So if you recall from some of our earlier videos, particularly when we talked about enzymes, we said that there are some proteins that sit around in the cell already made, already translated, but they are folded into an inactive shape, and they have to be activated in some way. And the most common way to activate a protein is to phosphorylate it. So we talked about this. Binding of a phosphate to an inactive enzyme, or really an inactive protein of any sort, might cause that enzyme or protein to fold into an active shape. So we phosphorylate it, and the next thing you know, it has changed shapes, and it can do its job. So even though we turned a gene on, made RNA, got it translated into protein, the protein still may not do its job until it's been activated. But it also turns out that when proteins are made, they don't last in the cell forever. Many proteins only last for a short period of time in the cell before they themselves get broken down and degraded. So proteins kind of have a half-life. Proteins are not permanent. They're destroyed, and we may have to reform them. So cells are continually replacing degraded proteins. But again, that requires turning the genes back on and transcription, translation, and the whole 9 yards. All right, so we've made it through all the steps of eukaryotic gene regulation, trying to figure out how would a cell know when to turn a gene on and all that kind of stuff. Recall that human cells have 22,000 genes, but we would never have all 22,000 genes turned on and expressed and producing protein at the same time. So how do scientists know which genes are turned on at what time? How do we begin to read or even see the sheet music that's being played out in these cells? Well, there's a research tool called a DNA microarray that allows us to study the sheet music that's going on in a cell. DNA microarrays are a research tool for determining which genes are active in a cell at a particular time. So in other words, we want to know which genes are on at a particular time. And these arrays are used to compare and contrast gene activity in different cell and tissue types. In other words, I could use the microarray to figure out which genes are turned on in the liver cell right now versus which genes are turned on right now in a skin cell or a kidney cell. We can compare the sheet music of two different cell types and see what notes do they have in common, what's similar about these cells, and what's different in terms of their gene expression patterns. And another very good use of this is we can identify genes associated with cancer. We can compare the gene expression patterns of a cancer cell with normal tissue and try to look for, well, which genes are turned on in the cancer cell that are not turned on in the regular cell, and things like that. So to give you to kind of go back to an earlier example, recall that in our liver cells and our lens cells of the eye, we have the exact same genes present, but different genes are turned on in those different tissue types. The liver cell had the albumin gene on and the crystalline gene off, whereas the lens cell had the albumin gene on and the crystalline gene off. DNA microarrays are the research tool that allows us to figure those types of things out. So let's take a look at how these DNA microarrays work. So the microarray itself is a little glass slide. It's about the size of a microscope slide. And it's got all these little gray spots on it. A DNA microarray actually has more spots than this, but I tried to make a figure where we could see them individually. So what is a spot? A spot, if we enlarge it here, is a place where we have attached a bunch of single-stranded pieces of DNA. And they are single-stranded DNA sequences that are unique to a specific gene. In other words, those 4 strands you see poking up there, they're all the same. All 4 of them are identical to each other. And they are a little stretch of nucleotide sequence that is unique to a specific gene. Let's pick an example. Let's say the hemoglobin gene. So that's a little region of DNA that we would find uniquely in the hemoglobin gene. Whereas this dot over here has got some pieces of DNA, single-stranded, that might be unique to the collagen gene. In other words, each little gray dot is a little probe to help identify a specific gene. Humans have 22,000 genes, so theoretically we could make a DNA microarray with 22,000 dots, one dot for each gene. So what do we do with this? Well, we start off with some samples. Let's say we want to compare the gene expression pattern of brain cells to the gene expression pattern of liver cells. We know that brain cells and liver cells possess the same genes because they all came from the fertilized egg originally. So they've got the exact same set of genes, but what we're asking is which genes are turned on and off in these different cells that accounts for their different phenotypes, their different appearances, and their different roles and functions. So we need to obtain some samples of brain cells and liver cells. Then, in a lab, we extract or isolate the messenger RNAs that are present. Now, messenger RNAs are only made when a gene is turned on. So when the gene is turned on, we transcribe it into messenger RNA. Okay, so now we've got these messenger RNA molecules that reflect which genes are expressed at the moment in those cell types. But RNA is very unstable. So the next thing we do is we reverse transcribe the messenger RNAs back into what is called cDNA. So we're just turning the messenger RNA sequence back into a DNA sequence. It's still reflective of which genes are turned on in those cells. We just have in the form of DNA again instead of RNA because the DNA is more stable. The next step is to label or color those DNA samples with fluorescent dyes. So maybe we'll label the brain cell DNAs green and the liver cell DNAs red. And again, these DNAs are just copies of the messenger RNAs. So again, it is reflective of which genes were on at the moment these samples were taken. Now that we have labeled these pieces of DNA with the fluorescent dye, we dump them onto the array, and these pieces are single-stranded, and what they're going to do is bind to their matching sequence that's already located on the array. Remember those little probes that were on there? So we dump them on there, and they will go find their little gene sequence that they're complementary to and stick to it. So what will this look like? When we evaluate this now, let's enlarge this. When we dump those pieces of nucleic acid on here, they'll go bind to their probe, their corresponding probe, and that will cause all these colored areas on there. And we get essentially 4 colors here. We've got gray, red, greens, and yellows. So what does all of that mean? Well, if it remains gray, that means that nothing in our samples, brain cells or liver cells, bound to that particular gene. The gene is present in the brain cell and the liver cell, but apparently those genes were not active. In other words, there were no messenger RNAs having been made of those genes that we're detecting here. So gray just means nothing bound. Those genes are off in both samples. Yellow, on the other hand, means that both the green sample and the red sample bound to the same probe location, telling us that whatever gene that little dot represents was expressed and turned on in both tissue types. When you combine red and green fluorescent coloring together, you get kind of a yellowish colors. Then, if you see a green dot, that's telling us that that particular gene was expressed in the brain cells, but not in the liver cells. And if you get a red dot, that's telling you that that particular gene, whatever it represents, had been turned on in the liver cells, but was not being played in the brain cells, at least at the moment that these samples were taken. So it really is a snapshot of the sheet music. This is telling you what notes are being played and what combinations with other notes in these particular cell types. And it allows us to compare the sheet music of the brain cells to the liver cells simultaneously. Now I'm showing you, a little cartoon version that I made here in PowerPoint, but here's a picture of an actual DNA microarray that could then be examined by a computer. So you see all these hundreds and hundreds of colored dots, greens and reds and yellows, and in this case black. Black would mean the same thing as gray, that no gene was turned on. And you might also notice there's some different intensities. Some of them are bright green, others are lighter green. It's not just an all or none thing. The amount of sample that binds will dictate the extent to which the gene is turned on. Some genes are turned on really strongly and other genes are expressed at a much lower level. So not only the color, but the intensity of the color tells us about the expression of that gene. So DNA microarrays have become an extremely valuable research tool. Now what I've got here is a picture of a tadpole. So now that we have discussed all this sheet music How does a cell know when to turn a gene on and what other genes will be turned on at the same time? The DNA microarrays can sort of reveal the sheet music. Here is the sheet music being played out. When the right genes get turned on in the right cells at the right time in the right combinations, something like this process begins to unfold. So various cells in that tadpole are starting to make different proteins. They're degrading proteins, producing new proteins, turning genes on, turning genes off. The sheet music is being played, the orchestration of genetic expression. And it's just fascinating to watch that orchestration of genetic programming play itself out. Now, this video there started with a tadpole, so a lot of cells had already become certain types of cells. Tadpoles already have muscle cells and skin cells. Let's back up a minute, though, and say, well, that had to have begun literally from a fertilized egg cell. It had to start out with a cell that just looked generic. And as that cell divided into 2, into 4, into 8, and so forth, it became many, many more types of cells. But those cells also had to differentiate. They all kind of came from that original fertilized egg cell, but eventually some have to really become muscle, some become skin, some become nerve cells, and so forth. And so that leads us to an interesting discussion of what we would call stem cells. So stem cells are undifferentiated cells with the potential to develop into a variety of different cell types. So the fertilized egg goes through a few cell divisions and it produces a ball of cells, but then those cells eventually have to commit themselves to becoming certain types of cells: nerve cell, muscle cell, skin cell, whatever. So when stem cells divide, first of all, they can produce more stem cells, or they can start to transition, start to play their own version of sheet music and become a precursor cell, the precursor or the beginnings of a specific cell type. And so they could eventually develop into these different types: fat cells, bone cells, white blood cells. Stem cells have the potential to become a wide variety of different cell types. Now, some cells can become any cell type, whereas other stem cells are limited. So let's discuss that for a second. Embryonic stem cells, the ones that are at these very early stages of the embryo, we can break that embryo apart, we can grow them in culture, and if we kind of splash on them or supply them with the right growth factors, the right cell signaling molecules, we can get them to turn on certain genes and begin the orchestration, playing the sheet music of various tissue and cell types. And embryonic cells, stem cells, can become any type of later cell type. And the term for that is totipotent. The embryonic stem cells are totally potent. They have the potential to become or develop into any type of cell. I used in the definition there the word develop. Really, the sciencey term would be differentiate. They are able to differentiate into any type of cell. So embryonic stem cells can become any type. Just got to get the right sheet music played in there, starting from scratch. But there's also something called adult stem cells. We can find those, for example, in bone marrow, and they can become a few types of cell types. Adult stem cells cannot become just any kind of cell. They're kind of limited into the range they can become. And the word for that is pluripotent. There are many, pluri like the word plural, so there are multiple types of cells that they can develop or differentiate into. So that's the difference between embryonic and adult stem cells. And these are fascinating types of cells to study, in part because we can learn a lot about gene regulation, but also because there's a lot of potential medical applications. You've probably heard about stem cells being used to treat things, or at least being studied for the possible treatment of things. The idea is that if you have a disease or a condition in the body where the cells are not functioning correctly, we could inject some stem cells into that region and let those grow into healthy versions of those cells. So what would be some possible stem cell applications? Well, let's take diabetes, for example. The cells in the pancreas are supposed to be producing insulin to help regulate blood sugar levels, But if the pancreatic cells are not functioning right, then maybe we could inject some stem cells into the pancreas that will develop into correct functioning pancreatic cells. So stem cells definitely offer some possibilities for treating diabetes. We could do something similar with heart disease. If heart cells have died off because of heart attacks, we could inject stem cells into the heart and let those stem cells differentiate into heart cells. We could even initiate the differentiation in the culture dish before we inject them. But the idea is inject these stem cells into the heart and they can grow into new healthy heart tissue to replace the damaged tissue. And something similar can be done with neurological disease. Things like Parkinson's disease and Alzheimer's could potentially be treated with stem cells. Or someone who has severed their spine in an accident and has become paralyzed. There's the possibility of injecting stem cells into that damaged region to replace or rejuvenate those cells. So there's an awful lot of possible medical applications with stem cells, and that's why there's a lot of research going on with them right now. Now, the last topic that I want us to get into in this video, another sort of scenario where the sheet music of cells playing out their genetic expression, is with cloning. Cloning is the artificial production of genetically identical cells or organisms. So cloning, believe it or not, has actually been around for a while, at least if we're talking about simpler types of cloning. In fact, the very first cloning of a vertebrate was done in 1902 by Hans Spemann. He cloned the first vertebrate really back at the turn of the previous century. It's pretty fascinating what he was able to do. The vertebrate that he cloned was a salamander. And let's look at the method that he used to do it, because it's a little different than the way we clone animals today. So back in 1902, he took a fertilized salamander egg. So a sperm has come along and fertilized the egg. And he let that go ahead and divide into 2 cells, as it normally would do. But at the 2-cell stage, under the microscope, he lassoed it with a little hair from a baby. He took a human baby hair and he wrapped it around the 2-cell stage of this developing embryo and used it like a little noose. And he pulled it tight and separated those 2 cells into individual cells. And once they had separated from each other, each one then grew into a salamander. But essentially, these 2 salamanders came from the same fertilized original egg cell. So these salamanders are clones of each other. And I just find it amazing what he did under the microscope with these little hairs that he plucked off the head of his daughter, who was a baby at the time. So he had this little fine little thread that he could wrap around and do this. So those were the first vertebrates cloned. Now, interestingly, he did that at the 2-cell stage. He repeated the experiment at the 4-cell stage and the 8-cell stage, and for the most part it worked even after they were larger clumps. But eventually, once you had a certain number of cells, it no longer worked. You couldn't take a blastula, for example, and wrap a noose around it and break it in half and hope that each clump of cells would become a new salamander. It only worked at these very, very early steps of cell division. It's almost like the very first fertilized egg cell was just beginning to play its sheet music. And once you had enough cells playing different sheet music, it just simply didn't work. You couldn't get the cells to back up and start the music over, so to speak. That was also done on a salamander, which is a type of amphibian. People thought it probably would be very difficult to clone a mammal, and that is why researchers and scientists were so shocked in 1996 when Ian Wilmot and a team of scientists announced that they had cloned a mammal, specifically a sheep, which they named Dolly. It kind of caught the research community by storm, in part because they didn't do it the way Hans Spemann had done it. They used a different technique using adult cells as the source of the genetic material. cells that had already differentiated into a cell type. They were able to use those cells to create the clone, and it just was amazing what they pulled off. So let's look at the cloning method that was used by Wilmot and his team to clone Dolly the sheep. They used a process called the nuclear transfer method of cloning. So here's what they did. They took a sheep, a black-faced sheep, as you can see there, and they took an egg out of its ovaries, and then they removed the nucleus from the egg. The term for that is an enucleated, the letter E, enucleated, meaning they removed the nucleus. So they've essentially got an egg cell lacking any genetic instructions. It's lacking the nucleus. Then they took another sheep, notice it has a white face, and they took a cell from its udder. So it's an adult cell, and they took this udder cell and they basically injected it into the empty egg with the idea that that donor cell nucleus could become the nucleus of the egg. Now let's back up a second. The nucleus removed from the egg would have been haploid. Remember, gametes are haploid. There would have been, one set of chromosomes. But they're putting in a diploid nucleus. So it's almost as if they're creating a fertilized egg by putting a nucleus in from an adult cell, from a different sheep for that matter. They then allowed that egg to go through some rounds of cell division to produce a blastocyst, and then they put that blastocyst into a surrogate sheep to carry it to term into a baby. And that surrogate mother gave birth to the clone that they named Dolly. Now, my first question for you is, which sheep is Dolly a clone of? Is it a clone of its surrogate mother that gave birth to it? Is it a clone of the sheep that donated the egg? Or is it a clone of the sheep that donated the nucleus? And if you look at this, hopefully you have figured out that Dolly is a clone of the sheep that donated the nucleus. And one of the reasons I pointed out the black face, white face, they intentionally used a black face sheep for the egg and a white face sheep for the nucleus donor because they wanted a very visual proof at the end of which sheep had been cloned. And Dolly, of course, is white-faced. And they intentionally used a black-faced sheep for the mother as well, so that when Dolly was born, they were expecting a white-faced sheep, and that would tell them that it truly was a clone of the one they thought it was. In fact, here's a picture of Dolly and Dolly's surrogate mother. So that's the sheep that gave birth to Dolly. Dolly has nothing genetically in common with the surrogate mother, or for that matter, really even the egg donor. Dolly is a genetic clone of the one that donated the essentially diploid fertilized nucleus. Now, Dolly is an interesting story. You see her dates there. She only lived to be about 7 and then died because the DNA that was used, it came from an adult sheep. So it was essentially mature adult DNA that became the starting material for all of Dolly's cells. And the donor sheep's DNA was already aged, so to speak. So it's almost as if Dolly was born with adult DNA. In other words, it's already acquired certain types of mutations. It had already degraded a certain amount. And yet that was the DNA that Dolly was starting her life with. So Dolly had a shortened life. She was then taxidermied, as you can see, and she's on display in a museum if you'd like to go see Dolly, so to speak. But that really surprised the world that they pulled that off, because no one thought you could take an adult nucleus and put it into an egg cell and expect the sheet music to start over from the very beginning. And in fact, the researchers knew the odds against it was pretty high, so they did several versions of the experiment. Hundreds and hundreds of egg cells were isolated and nuclei put in them, and the majority of those outcomes did not work. They did not produce a baby sheep. They produced little balls of kind of odd tissue with teeth and fur and just crazy things, none of which could be carried to term. But they did have this one where it worked. And once they knew which one worked, they could reverse engineer what conditions allowed it to work. So cloning has actually become quite common now. And many, many, many different types of animals have been cloned. You see the pigs here have been cloned, cows have been cloned, horses have been cloned, dogs and cats and rabbits and monkeys. Just all kinds of things have been cloned now that we know how to do it. And one advantage of cloning that could be argued is just take your best cow with the most meat or the best milk producing or something and just clone it over and over, so you always have the best sources of food or milk or dairy products, or the strongest horses or whatever you want. You just clone the best ones. Now, whether we should do that, I don't know, but that's one of the arguments for cloning. In fact, one of them that's definitely a little bit controversial, these are some rhesus monkeys that were cloned, but what they did is they had used CRISPR, a gene editing tool, to create a monkey that had some sleep disorders, but it also had some anxiety and schizophrenia. But they found this, they'd created this monkey that had these human-like diseases, and then they cloned it so that we now would have lots of monkeys that we could use as medical models for studying these human conditions of sleep disorders, but also anxiety and schizophrenia. And that raises a very interesting ethical question. Should we be, first of all, creating those disorders and then cloning them into other primates like this, much less other animal types? Now, another interesting use of cloning is to possibly resurrect extinct species. These are the remains of a woolly mammoth that were preserved well enough that they were able to get some intact DNA samples from it. And the thinking was, can we take the mammoth DNA we have, even though it's from adult cells, could we put those into an empty elephant egg cell? Take an egg cell from an elephant, remove the nucleus, and then put a mammoth nucleus in it, essentially, and put that in a surrogate elephant mother and see if the mother can give birth to a mammoth. Can we resurrect the mammoth by these cloning techniques? And that is a process that is still being tried to succeed. The DNA is damaged a bit, plus an elephant is not really the same thing as a mammoth. So there's some slightly different chemical compositions to the egg that comes from an elephant and so forth. But they're working on it. And at some point, we may be able to resurrect some other extinct species. Some people think we might be able to resurrect some dinosaurs using large birds as surrogates and stuff like that. So some interesting stuff. And so finally, what that does is that tells us there's two types of cloning we could think about. There's what's called reproductive cloning. That's what we've just been discussing. The use of cloning techniques to produce complete adult cloned organisms, such as Dolly, or certain livestock animals or mammoths, something like that. And then there is therapeutic cloning. So that takes us back to the stem cell story. We can use cloning methods to create pluripotent and totipotent stem cells. And then we could use those clone cells for therapeutic purposes, such as treating cancer or diabetes or skin grafts or nerve damage or all sorts of options. Either way, whether we're talking about reproductive cloning or therapeutic cloning or stem cells in general, clearly there are a lot of bioethical questions to be addressed. And that is beyond the scope of what we can get into in this course. But I hope it is raising all sorts of interesting questions in your mind about what types of techniques ought we be doing, which ones should we not be doing, which ones might simply require more oversight or regulation. Just a lot of really complex questions that we're having to ask, because our ability to do these things, our understanding of these processes is just increasing at a rapid rate. So we need a lot of people studying not only the biology of all this, but also studying the philosophy and the theological pieces and just the ethics of it all. So just a whole nother area of topic we could get into, but we won't. In fact, that brings us to the end of this video on gene regulation. A lot of interesting stuff going on in that video. A lot of different research techniques and so forth. So I hope you found some of that stuff interesting. And we will get into some more interesting things in our next video. So we'll see you then.